I spoke in this seminar a couple years ago about a method we developed to phase and impute repeat polymorphisms in the genome. I have assumed that most of you either weren't here or don't remember much from that, so there may be some overlap with that previous talk, but I hope I don't bore you too much for that. So yeah, I'm excited to tell you today about our work on imputing repeat polymorphisms and the role that protein coding repeat polymorphisms in particular play in driving genetic associations. Almost all of the work I'll describe here is detailed in our recent bioRxiv preprint that we posted in January. So if you want any details, please see that. Okay. So the starting motivation for this is that there happen to be hundreds of proteins scattered throughout the genome that exhibit heritable size variation due to coding repeat polymorphisms. An instructive example of this is the protein lipoprotein a blood lipid of mysterious function whose concentration is in blood is an important risk factor for cardiovascular diseases. Lipoprotein A consists of an ApoB100 molecule, depicted here in red, that binds to cholesterol, and an ApoA molecule, which consists of a series of Kringle-shaped K domains. The number of Kringle IV2, a particular type of this domain, varies from particle to particle and person to person. And the number of KIV2 domains is determined genetically by the number of variable tandem repeats, the variable number of random repeats of a domain in the gene LPA encoding this ApoA molecule. So VNTR is a genomic region where a nucleotide sequence is repeated a number of times, which varies commonly and widely among individuals. In this fish microscopy image of LPA alleles, you can see two LPA alleles, one with 4 copies of the KIV2 domain and another with 20 copies of the KIV2 domain. Can you guys see when I use my pointer?
Yeah.
Yeah.
Okay, great, good. So the 5.5 kilobase repeat unit spans 2 LPA exons encoding a combined 114 amino acids, exactly one of these little Kringle domains. So after transcription and translation, the top allele will produce a small ApoA isoform with 4 KIV2 domains, and the bottom allele will produce a large ApoA isoform with 20 KIV2 domains. Since VNTRs are difficult to measure directly from SNP array and sequencing technologies used in most genetic association studies, the extent of this variation and its phenotypic consequence are largely unknown. That's the question we asked in this project. In this project I'll tell you about today, we attempted to survey the landscape of coding VNTR variation and determine the role that coding VNTRs play in the inheritance of complex human phenotypes. Our goal was to leverage available biobank scale genotype and phenotype data to survey this landscape. In particular, we were motivated to use the UK Biobank, which collected extensive phenotype and genotype measurements for approximately 500,000 participants. Hopefully these are some reminders to this audience, but most of the participants are white Europeans with about 10,000 participants from other continental ancestries. Phenotype information was collected from a variety of sources, including participant surveys, linked electronic health records, and measurements from blood samples. All participants were genotyped on one of 2 SNP arrays, and additionally, around the time we began this project, whole-exome sequencing data became available for about 10% of the cohort, or 50,000 individuals. Recently, they expanded the whole-exome sequencing cohort to 200,000 individuals. The challenge for studying VNTRs in the UK Biobank, as I mentioned, is that VNTR allele lengths are not easily measured from SNP arrays or exome sequencing. So backing up, let me tell you a bit about genotyping VNTRs. Traditionally, there were some low throughput methods that directly measured VNTR allele lengths in small cohorts, maybe hundreds to maybe a couple thousand individuals. Example technologies include gel electrophoresis, quantitative PCR, and the fluorescence imaging that I showed you on that first slide. This histogram down here on the right is from a 1992 gel electrophoresis survey of KIV2 VNTR alleles in 156 individuals. When we began the project, the state-of-the-art methods estimated VNTR lengths from whole genome sequencing data. And these methods kind of broadly fell into two classes. One class of methods directly genotype VNTR alleles from spanning sequencing reads. This class of methods can accurately genotype short VNTR alleles, say up to 100 or 150 base pairs long. An obvious limitation of this class of methods is that VNTR alleles longer than an individual read cannot be distinguished from one another. As long read data becomes cheaper, more accurate, and readily available, this approach will become more attractive and maybe the way to go when trying to understand VNTRs. Another class of methods tries to estimate VNTR lengths from whole genome sequencing depths of coverage. If you randomly and uniformly cut up and sequence an individual's genome, the amount of DNA sequence from a particular VNTR should be proportional to the length of that VNTR. By measuring the number of reads that actually map to the VNTR, the so-called depth of coverage at the VNTR, you can get an estimate of the VNTR length. This is a figure from Hansaker et al.'s 2015 Nature Genetics paper, which detailed a method to estimate VNTR length by normalizing depth of coverage signal in whole genome sequencing data. Those authors demonstrated that their method, which they call genome strip, is accurate and precise at certain genomic loci, including the LPA KIV2 repeat. But this method works best when you have a lot of reads and really at the multi-kilobase scale at the very large end of the VNTR spectrum. So summarizing, the existing methods for measuring VNTRs and sequencing data are good at measuring very short alleles with spanning reads or very long alleles via depth of coverage in whole genome sequencing data. There are two serious obstacles to using these methods to study VNTRs in the UK Biobank. First, there's this big range of intermediate VNTR allele lengths that aren't really accessible to these methods. They're too long to be spanned by short reads and and not long enough to be accurately estimated from depth of coverage. Second, whole genome sequencing data is not available at the UK Biobank yet. The main difference between whole exome and whole genome sequencing data is in read capture to target the exome, a process which can introduce noise and bias in depth of coverage estimates for VNTR lengths. So to address these gaps, we made a couple of innovations. First, we developed methods to estimate VNTR lengths from whole exome sequencing depth of coverage. And second, we developed methods to model VNTR haplotype phase and sharing. By modeling haplotype sharing, we can pool information from noisy measurements across individuals who inherited the same VNTR allele identically by descent from a recent common ancestor. Employing these methods, it refines our VNTR measurements and allow us to probe the effects of VNTR alleles at shorter length scales and can be accurately estimated from individual samples alone. So I'll tell you a bit about both of these sets of methodological developments now. So we found that particularly in the UK Biobank, because of the uniform and high coverage whole exome sequencing conducted by the UK Biobank, that really helped us call VNTR lengths, estimate VNTR lengths in their participants. The details are a bit technical, but in broad strokes, we estimated VNTR lengths summed across the two parental alleles from depth of coverage and normalized the depth of coverage against samples with similar coverage profiles to reduce technical noise. In some instances, this normalization against other similar samples reduced the noise in these measurements by up to 60%. We benchmarked these methods using data on siblings. At LPA, with VNTR alleles ranging from 10 to 20 kilobases long, siblings that were IBD2, that is, inherited both alleles identically by descent from their parents, had a 93% correlation in their measurements. At MUC1, a shorter VNTR with alleles ranging from 500 to 5,000 base pairs, IBD2 siblings had 80% correlation in their measurements. Noisier than LPA, but still a good amount of signal here. Okay, so that's estimating going from a whole genome sequencing to whole exome sequencing. What about using haplotype sharing to kind of clean up these noisy estimates? So the methods we developed to model VNTR haplotype phase are adapted from the Lee and Stevens model for phasing SNPs. So I'm going to start by reviewing that a bit. The heart of the Lee and Stevens approach to phasing SNPs is a generative model for constructing new haploid and diploid genomes from a reference panel of known haplotypes. So given a panel of haplotypes, Lee and Stevens define a probability distribution over new haplotypes. You can think of a haplotype as a sequence of zeros and ones corresponding to the reference and alternate allele at each SNP. A typical new haplotype is a mosaic built out of long contiguous stretches of reference haplotypes, like this, with a small number of jumps between panel entries. The jumps are meant to model recombination events, and the model's preference for contiguous stretches of reference haplotypes encodes the local correlations and linkage disequilibrium we see in SNPs. The precise model, which gives its probability distribution over haplotypes given the panel, is specified by a hidden Markov model, where the hidden variable is the shared or copied haplotype at a given location in the genome. Okay, so that's to generate a haploid genome. To generate a diploid genome, you simply sample two haploid genomes independently. Okay, so that's the Lee-Stevens model for SNPs. How do we go to VNTRs? Well, this model extends pretty easily and naturally to VNTRs. So to generate a new haploid genome from a panel of known SNP and VNTR haplotypes, we simply generate a mosaic of copied haplotypes from the panel as an ordinary Lee-Stevens SNP HMM. The SNPs are generated in literally the exact same way as in the ordinary Lee and Stevens model. The VNTR length is drawn from a normal distribution whose mean is the length of the allele of the copied haplotype as defined by the HMM hidden variable. So in this instance, if we're making this red haplotype, at the VNTR, the red haplotype is copied from the reference haplotype I. So we're going to draw the length of the allele from a normal distribution centered at the length of haplotype I's VNTR.
I think you just answered my question, Ronen.
Okay.
But so right, in ordinary Lee-Stevens, it's not just a mosaic, but importantly, it's an imperfect mosaic, which and those little those little errors allow for both sequencing errors and de novo mutation.
Yes.
So here you've done the same thing for the length as well. You've allowed it to move around a little bit in the copying, right?
That's correct, and perhaps have a different mutation parameter than the what you'd allow for the SNPs.
Sure. Yeah. And you learn that, or?
And yeah. So the width of this distribution is learned from the data,
yeah,
and meant to allow for error in the panel VNTR lengths and the possibility of mutation between, the copied haplotype is supposed to be the haplotype with a recent common ancestor,
yeah,
with the new haplotype, but perhaps there was a mutation between.
Right. Anywhere between the common ancestor and either of the two, the one in the
that's right. Yeah.
Okay. Cool.
Okay. Great. Thanks. Other questions? Okay, great. So this gives a probability distribution over SNP haplotypes and lengths given a panel of reference haplotypes. And as before, we can generate a diploid genome by sampling two haploid genomes independently. Okay, so how does this help with our phasing? Well, so what do we have? So suppose we have this panel of phased SNP and VNTR haplotypes, and now we come along with an individual with known SNP haplotypes and VNTR lengths estimated from depth of coverage. I'm going to assume the SNPs have been phased because actually, that's been done in the UK Biobank already. So all we have to do is phase the VNTR length, take the diploid measurement of the length summed across the two alleles and split it into the contribution from each allele. So as I said, the VNTR length is a potentially noisy estimate of the sum of the lengths of the two parental alleles. So the generative model dictates a posterior distribution over the individual allele lengths, L and L prime, conditioned on the SNP haplotypes H and H primed, and this diploid measurement, which is possibly noise. So L plus L prime plus some measurement noise, and of course the panel. Taking expectation yields estimates for the VNTR lengths at each allele. These estimates somehow incorporate haplotype sharing information, in effect pooling noisy depth of coverage estimates across many individuals. The expected VNTR allele lengths can be computed efficiently using HMM techniques. The generative model also dictates a posterior distribution for individuals with SNP haplotypes only, allowing us to impute VNTR alleles into SNP array data, which we'll do later. We'll phase the exome sequenced individuals in the UK Biobank and impute into the participants who were only SNP genotype, SNP array genotypes, non-sequenced.
Ronen, I got a quick question. So
yes, please.
What is the sort of the distribution? Okay, so the typical length between two SNPs that goes into the panel and one of these repeat units. Like, do you have like SNPs inside these repeat units, or the SNPs are sort of far and few in between them?
That's a great question. So we specifically excluded SNPs within the repeat units for this portion of the analysis. SNPs that are within the repeat units are quite difficult to call accurately. I mean, because you can imagine if you have 10, this happens all the time actually at LPA. You have a 5 kilobase repeat unit, and maybe you have 10, 20 copies of it, and 3 copies contain some common variant. And then you see kind of a mess in the reads.
But you probably don't get a common polymorphism like that inside very often. Like the SNPs you're talking about here are the common polymorphisms that are used to identify the haplotypes, which are a bit sparser, right?
Well, I mean, there is common variation within the repeat units, which I've elided this so far.
Okay.
This is a detail I wasn't going to go into. We thought very carefully about this because that kind of thing can actually bias your whole exome sequencing estimates, and we did a lot of work to try to correct for that. And so, for instance, for LPA, there are three very common types of repeat units. And that's not an LP. I mean, LPA's got these huge repeat units, so you might think, well, it's just because they're so huge that this happens. There are other VNTRs that came up in our study had similar variation within repeat units.
Interesting.
Yeah. Okay, so to answer Meritosh's question, we avoided all of the SNPs at the repeat unit. We just cut them out of this analysis to avoid this issue. And then in follow-up analysis, we studied the variation within the repeat units pretty extensively. Okay. Right. So we can now phase and impute VNTR alleles. Something that's a little bit technical, if this doesn't make sense, don't worry about it. But we don't really start with a reference panel. All we have is kind of a panel of SNP haplotypes, and then these exome sequenced diploid measurements of VNTR lengths. So for a cohort with depth of coverage estimates only and no panel, we applied the leave one out framework that is usually employed in Lien-Stevens SNP phasing, namely in this strategy, you start with a randomly chosen phase for the entire cohort and then cycle through the individuals in the cohort, rephasing each one individually using the rest of the cohort as a panel. The hope is that you converge to a sensible answer, and specifically the strategy is iteratively seeking to maximize the so-called product of approximate conditionals. Anyhow, after extensive prototyping and benchmarking this method, we discovered that the HMM mosaic copying probabilities seem to sort of overweight the best matches in the panel, and that we could achieve similar or improved results even by replacing the copying probabilities with simpler scores computed from identical by state length between haplotypes. Since this approach had advantages in runtime and parameter tuning, we actually adopted this simpler approach for our analysis in UKB. So how did this help? So as I promised, this haplotype phasing and haplotype sharing should refine the VNTR length estimates. At LPA, estimates, our estimated R squared to the truth for our whole exome sequence estimates only before phasing had about 0.9, R squared of 0.93 to the truth, which improved to about 0.97 after phasing. At MUC1, kind of a medium-length VNTR, as I mentioned, the R squared to the truth was about 0.79, which jumped to 0.95 after incorporating all of this haplotype sharing. And at this very short VNTR at TENT5A, we started with an estimated R squared to the truth of 0.66 and jumped to 0.83 after phasing and incorporating haplotype sharing. So this really does give a boost, at least in certain loci, to your ability to estimate VNTR lengths. Okay, so that's what I wanted to say about the methods. I'll now tell you a bit about our survey. So we applied these methods to survey approximately 10,000 repetitive coding loci in the UK Biobank, identified by the software tandem repeats finder. And this is kind of how it worked out in numbers. So we start with 10,000 repetitive coding loci, the vast majority of which are probably not VNTRs. So we estimated their depth, their lengths from depth of coverage, and then phased and imputed these loci. And then we applied stringent quality control measures to, for instance, ensure that the locus exhibited heritable length variation and was not part of a larger CNV. This left us with 118 likely coding VNTRs that were amenable to our methods, which we tested against 791 traits measured by the UK Biobank. We found 180 of those VNTR phenotype pairs passed a stringent genome-wide significance threshold for SNPs, 5 × 10⁻⁸. And then we ran the software find map to compare the association statistics of the VNTRs at these 180 significant associations with those of nearby SNPs and determine which of those associations appeared to be driven by the VNTR rather than the SNPs. This left us with 5 VNTRs and 19 associations, which follow-up analyses confirmed that the loci appeared to, that the VNTR appeared to be causal at the locus. In most of the instances, the VNTR had the strongest association at the locus, beating all nearby SNPs and indels. Find mapping is just a little bit easier in this situation because the variants are so multi-allelic. They are generally not well tagged by the nearby biallelic variants. And so these are the associations we found. So here I've listed the gene that's affected by the repeat polymorphism, the repeat unit size, the number of repeats found in a typical allele, the domain affected, the associated phenotype, the main associated phenotype, the effect range between the longest and shortest allele, and the p-value for the association. And you'll notice that by far the strongest association in our data replicated the known association between the LPA KIV2 repeat and lipoprotein A concentration. The 4 other gene phenotype pairs, as far as we are aware, this work is the first to implicate the VNTRs at each of these associations. Some of the loci had been, like ACAN was a known locus for height, but the VNTR had not been implicated there. At ACAN, TENT5A, and MUC1, the VNTR association was actually stronger than that of any nearby SNP. So we further boosting our confidence that FindMap was doing something reasonable. So here are Manhattan plots for ACAN, MUC1, and TENT5A, and you'll see the VNTR beats all the nearby SNPs. At TCHH, the story is a little more complicated. There's a missense SNP identified in the hair curl literature that slightly edged out the VNTR in association strength. This SNP is not an LD with a TCHH VNTR, in fact, the VNTR was not well tagged by any SNPs. And after conditioning on the SNP, the TCH VNTR remained significant and was the top association at the locus. So
there's a question in the chat by Yossi. He asked, what % of the variance is explained by these variants?
Good question. Take with a grain of salt because I'm recalling them from memory. ACAN, it's about, these are very polygenic traits. At least height and urea are very polygenic traits. So any one locus is not going to explain a ton of variance. I think in Europeans, the ACAN VNTR explains about 0.3% of variance.
For a single locus, that's like absolutely huge.
Yeah, it's large for height. Yeah.
Yeah, sorry, that's what I meant, for height.
Yeah. Yeah, I think to people who aren't in GWAS, that, maybe that, or maybe a poor pure mathematician who's just starting in genetics, it sounds a little small, but it's pretty large for height.
I guess sorry, I'll jump in with voice. I guess it's confusing because the previous slide says 3 centimeters.
Yes. Okay. Great.
Now 3 centimeters is not 0.3%. So could you?
Well, the effect size is not enough to determine the amount of variation explained. You have to know the allele frequency as well.
Right. So, okay. Great.
So the point is that the common alleles have a much narrower range of effects. There are at the end of the talk, I'll pull up a figure on this. I wasn't going to talk much in depth about ACAN, but I'm happy to talk about it after. But the ACAN alleles range from 13 to 44 in Europeans, but almost everyone has a 26, 27, or 28 copy allele.
That makes a lot of sense. So the extremes are relatively rare, and so you don't get much population variance explained, but there's enough deviation to reliably infer that it has a large effect size.
Yes, that's right.
I guess I know maybe this table is already busy enough, but I think it would be a useful addition to say the kind of I don't know, some sort of number on tip, variance. What is the variance that is something to
or some kind of indication of the distribution of the repeat count and not just the range.
Yeah.
Right.
Yes. Thanks for those suggestions. I think we thought well, I'm glad you guys are impressed by that number, the amount of variance explained. We thought that maybe for some of our readers, the effect range would be a little more striking. We thought so that's what we included in this table. But and for this crowd, maybe I should have generated a different table, but I was lazy and copy and pasted from the paper. But those are important points. Yeah. Well.
So is the 3.4 actually just coming from a linear assumption about the effect of the number of repeats plus the inferred kind of slope, the effect size? One additional.
It's inferred from binning alleles based on a copy number.
Okay.
Maybe since there are so many questions, if I exit out of this, I think I'm gonna. Basically, we measured the effect of a 13 allele and a 44 allele. And we saw that, the average person with a 13 allele was 3.4 centimeters shorter than the average person with a 44 centimeter allele, properly conditioning on PCs.
Well, you really did see that despite the rarity of such extremes, you really did observe that for those people in the extremes. It's not just an extrapolation from what happens.
Yes, and that's right. And
that's basically my question. That is more convincing. Okay.
Yeah. I'll show you the figure at the end of the talk if you ask again.
Thanks.
Other questions? Well, I should say, since the amount of variance explained came up, after seeing this plot and looking at the hair curl literature, which had this eye-popping figure for this missense snippet, I think it explains something like 5 or 6% of variance in hair curl, which is not measured in UK Biobank. We decided to impute the TCHH VNTR into a data set that had hair curl, namely the Twins UK. And, we were hoping we'd get an eye-popping number like that for the VNTR as well. It turned out it was lower than the missense SNP, but still the second most, after conditioning on the missense SNP, the strongest association in the genome with hair curl. Okay, so continuing on, just comparing these to other loci, the VNTR associations at MUC1 and ACAN were among the strongest in the genome. The MUC1 VNTR's association strength tied with a SNP on chromosome 5 for the top urea association in the genome. These are actually crazily close. These two peaks. ACAN was one of the first reported loci for Hyde appearing in a GWAS from 2008, and the VNTR is among the top height variants in the genome for Europeans, maybe in the top 10 or 15. Interestingly, in Africans, the association replicated strongly, and in fact beat every SNP in the genome. And this gets back to that previous discussion. First of all, we had way fewer African samples. There's like 7,000 African samples in the UK Biobank. Nonetheless, we were able to replicate this result. The reason is that unlike in Europeans, the distribution of alleles is much wider in Africans. And so actually the ACAN VNTR explains almost twice as much variance in Africans, like 0.6% of variance in Africans. Okay, so summarizing this little bit of the talk, we can estimate VNTR lengths summed across the two parental alleles from whole exome sequence depth of coverage. We can clean up these measurements by modeling haplotype sharing among individuals, refining the measurements, inferring individual allele lengths, and impute VNTR lengths into SNP array data, and coding VNTRs at LPA, ACAN, MUC1, TENT5A, and TCHH are strongly linked to diverse phenotypes. The ACAN and height and MUC1 and urea associations are among the strongest associations for these phenotypes genome-wide. Okay, so now I think I'm going to spend the rest of the time talking about what I think is really cool application of these estimates, which is a long story dissecting the genetic architecture of lipoprotein A concentration, which sometimes is confusingly called LPA, which is hard to disambiguate from the gene LPA. And I will try to specify, but if it's confusing, please let me know. So LPA, lipoprotein A concentration in the blood, is a highly heritable risk factor for cardiovascular disease. It varies more than 1,000-fold among healthy individuals and is extremely heritable and basically monogenic. The sibling correlation is 92% for siblings that are IBD2 at the gene LPA, which is, I'll remind you, the gene that encodes the ApoA part of the lipoprotein A particle. The function of lipoprotein A is a bit mysterious. Lipoprotein A concentration is a documented risk factor for cardiovascular disease and has been shown to associate with type 2 diabetes risk. The KIV2 allele length, the VNTR allele length, is the main genetic determinant of lipoprotein A concentration. As I mentioned, the KIV2 VNTR allele length determines the translated ApoA isoform size. In vitro studies have demonstrated that ApoA size in turn affects endoplasmic reticulum retention in the hepatocytes that manufacture ApoA. As a result, larger isoforms are secreted less efficiently from hepatocytes and result in decreased lipoprotein A concentration. I think an instructive analogy here, which since Yossi is in the audience, I got from Yossi basically, is with screws. If you have a short screw, it's easy to drive through a piece of wood because the thread length is short and there's not much friction. If you have a long screw, it's much harder to drive through wood because the long thread creates a lot of friction. Similarly, a small ApoA molecule has short thread, travels quickly through the ER and gets into the blood. Whereas larger molecules are retained in the ER longer and result in decreased secretion and decreased lipoprotein A concentration. From this effect alone, KIV2 length explains about 50% of lipoprotein A variants. So the starting point of any study of lipoprotein A in the UK Biobank, or maybe lipoprotein A in general, requires detailed knowledge of these KIV2 lengths, which are really explaining a lot of the variants. I'll remind you, lipoprotein A concentration is about 92% heritable. And so KIV2 length explains most of the heritability, but it's far from the entire story. So next we sought to identify the other genetic determinants of lipoprotein A concentration. So we had a first look at KIV2 in 1 million haplotypes. Here's the KIV2 allele distribution and the KIV2 by LPA scatter plot, UK Biobank. There are error bars in this plot, kind of unbelievably. Okay. So a key feature of the gene LPA that we exploited in our analysis is that there are seemingly healthy individuals with no functional copies of LPA. In particular, there are null LPA variants that result in null lipoprotein A concentration that have arisen to low frequency in the population. Here I've highlighted homozygotes and compound heterozygotes for 2 SNPs that we identified as resulting in null LPA in the literature. Were identified as resulting in null LPA in the literature. One is a canonical splice donor identified by Orel-Kova et al. In the 1990s. The other is a missense variant discovered last year. So the homozygotes and compound heterozygotes are highlighted in orange, and you see that 90% of the homozygotes and compound heterozygotes, the lipoprotein A concentration measurement was below 3.7 nanomoles per liter, the minimum reported value in the UK Biobank. Together, these SNPs rise to about 4.7% allele frequency in Europeans. So kind of really cool feature of this story is that carriers of these null LPA variants allowed us to measure lipoprotein A concentration contributions from individual haplotypes, which was particularly useful for downstream fine mapping of lipoprotein A. So in the top panel of this figure, we have the ordinary LPA KIV2 scatter plot with no restriction on the opposite haplotype. In the bottom panel, it's the same picture, but I've restricted to haplotypes that are opposite another haplotype that carries one of those 2 null alleles I just showed you. The restriction of the alleles greatly clarifies the picture of the KIV2 LPA relationship. You can see all of these points that had long KIV2 repeat and high LPA have just disappeared from this figure. And that's because in these points, the LPA is presumably contributed by the opposing haplotype. Whereas in these points, the opposing haplotype has contributed no LPA because it has one of these null variants. I should mention, I downsample this figure is the same as this ordinary scatter plot. I've just downsampled it so the number of points match. So I'm not biasing you with the number of points or anything. Okay, where are we? So by restricting to carrier, these guys, the carriers of the null variants, we are able to, in effect, analyze lipoprotein A concentration in a haploid model, an option exploited often by yeast geneticists, but usually unavailable to human geneticists. Okay, so staring at this figure, you might see that there's this prominent cluster of haplotypes with short and medium KIV2 length, but low LPA. Given the high heritability of lipoprotein A concentration, additional genetic variation must explain this low LPA produced by these alleles. So we next sought to identify these variants. To do so, we performed a stepwise conditional analysis to identify large effect lipoprotein A concentration lowering variants that result in low LPA, despite occurring on haplotypes with short KIV2 domains. At each step of this analysis, we performed a Fisher exact test on the binarized phenotype. And this is what happened in the first iteration. So here's a Manhattan plot describing the association statistics for each variant within 1 megabase of the gene LPA. It's a Fisher exact association statistic with this binarized LPA phenotype, restricting to short KV2. And the clear top variant turns out to be a cryptic splice donor internal to the KIV2 domain. This variant occurs at the last base pair on one of the 2 exons spanned by the KIV2 repeat unit, and when it's discovered by Coason et al. In 2017, with some wet lab validation to show that it affects splicing. The variant also gets some support from Splice AI, a deep learning predictor for the effect of variants on splicing. On the right, I've highlighted in orange the carriers of this variant, and you can see it lights up this cluster that we were trying to identify. So we did this in steps. I'll show you the first 3 iterations of our stepwise analysis. The first one we've just discussed, identify this cryptic splice donor with minor allele frequency 12%. The second top variant after excluding carriers of iteration 1 variant was this cryptic splice acceptor with Splice AI support and minor allele frequency 0.19. And then the third, after excluding both of these variants, the third top variant was canonical splice donor, that SNP discovered by Ogorelkova et al. In the 90s that we've been using to create our haploid model for lipoprotein A. Each of 12 rounds of this conditional analysis identified a large effect missense or splice altering variant that appeared to greatly reduce LPA. In each round of the analysis, the identified variant had literally the strongest association statistic among all variants within 1 megabase of LPA. In each panel of this figure, I've highlighted the haplotypes carrying one of these variants. These are the top 3 I just showed you, the 2 splice variants in the KV2 region and the Ogorelkova slip. So 6 of these are missense and 6 are splice altering. 6 of them some mix of missense and splice altering also had literature support for their causal effect on lipoprotein A concentration. And together these variants cover about 40% of European alleles, meaning that about 40% of European alleles are sort of broken and produce little or no LPA. And some additional fine mapping analysis that identified a couple of other probably causal variants, 5 rare null LPA variants and 3 common small effect variants that seem to exert a small consistent effect on lipoprotein A concentration across the KIV2 spectrum. All of these variants were either missense predicted to alter splice, stop gains, or in the 5' UTR. Having identified all these other sequence variants affecting lipoprotein A concentration, we can revisit the KIV2 relationship with lipoprotein A concentration. So after removing the, am I going to, sorry, Maritosh, am I going to 1150 or can I go a few minutes longer?
You can go a few minutes longer.
Okay, great, thank you.
Yeah.
I'll try to wrap up by 1155.
Sounds great.
After removing the 40% of the haplotypes that were sort of broken, this kind of weird, kinky LPA KIV2 relationship becomes much cleaner and is well modeled by a simple exponential. So in this figure, the haplotypes containing no LPA modifying variants are in gray, and those carrying a small effect variant are in green, blue, or red. The large data points represent phenotypic means for alleles binned across by KIV2 length and carrier status for the red, blue, and green variants. And the exponential relationship is really consistent across these variants, with the green, blue, and red variants contributing an additional multiplicative factor. Zooming out a bit, I purposely hid part of the plot in that figure. This effect seems consistent across the medium and long KIV2 alleles greater than 11 repeats, and seem to attenuate for alleles shorter than 11 repeat units. and even reversed for very short alleles. Biologically, we're not quite sure what's going on here, but statistically, the signal is very strong. There was a positive correlation between KIV2 length and lipoprotein A concentration in this very short KIV2 allele length regime with p-value of 5.1 times 10 to the minus 19. Combining all of these variants and KIV2 and all that we've learned about KIV2 into a model for lipoprotein A concentration explains 83% of the phenotypic variance and 90% of heritability. To estimate the contribution of a single haplotype to LPA, we constructed a haploid model, parametrically fitting the LPA KIV2 curve and allowing the additional fine map variance to act multiplicatively on top of the value predicted by the KIV2 length. To estimate an individual's total LPA, we simply added the contributions of the two haplotypes. By comparison, if you threw all of these variants into a standard model that's linear, standard additive model that's linear in SNP and VNTR dosages, you only explain 60% of variance, which is similar to the performance you get in recently published polygenic risk scores for this trait. Okay, to wrap this story up, I'm going to highlight a couple of applications of all this knowledge we've accumulated about LPA. And we can revisit some of the outstanding questions about lipoprotein A concentration. So it's been known for a long time that the KIV2 allele distribution and LPA distributions vary greatly across populations. Here's the LPA and KIV2 distributions for African, European, and Asian populations from a survey by Kraft et al. In the 90s. You can see that the median African LPA is much higher than the median Caucasian LPA, despite the VNTR allele, the KIV2 allele distributions being rather similar. Their medians are actually quite close to each other. So a big mystery was what explains this. Somehow, it was unclear if additional genetic variation accounted for these population differences in observed lipoprotein A, or if LPA just behaved differently in different populations because of unidentified environmental factors. And we found that our model really accounted for this variation. So here, for each of 4 continental populations, I've plotted median LPA from direct measurement in orange, from our genetic predictor for LPA in blue, and then using predicted from KIV2 only in gray. And as you can see, there's this huge difference in measured European LPA and in our genetic predictions for LPA among Europeans and Africans, but the KIV2 alone really can't account for this. And the explanation is that if you look at the allele histograms of KIV2 alleles in each of these populations, the allele histograms for the Europeans and Africans are really quite similar. But in this plot, we've grayed out the alleles in Europeans that are broken, that have one of these LPA reducing variants, and there are way more of them in Europeans and Africans, reducing the median LPA. Similarly, there's this LPA increasing promoter variant in red, which is much more prevalent in Africans, further adding to this disparity. Okay, maybe I will skip the cardiovascular diseases and tell you a last little thing about type 2 diabetes. So using our genetic prediction of lipoprotein A, we also were able to gain some insight into the known association between lipoprotein A concentration and type 2 diabetes. So there have been conflicting recent reports as to whether the low LPA is a causal risk factor for type 2 diabetes. The answer to this question is particularly pressing, as there are now LPA lowering therapies in clinical trials. So the association between lipoprotein A concentration is readily apparent, and type 2 diabetes is readily apparent in UKB. You see increased prevalence of type 2 diabetes for people with measured LPA below 10. But that association completely goes away when you turn to predicted LPA from genetics. So this indicates that there's something about type 2 diabetes that's lowering your LPA and not the other way around. So either type 2 diabetes itself, a comorbidity like liver disease, or medication used by patients with type 2 diabetes is lowering their LPA, not lower LPA causing their type 2 diabetes. Okay, to summarize, stepwise conditional analysis in an effective haploid model for LPA accounting for KIV2 length pointed to a series of about 20 LPA missense splice altering and UTR variants causally affecting lipoprotein A concentration. A haplotype-aware genetic model for lipoprotein A concentration explained 83% of variance and 93% of heritability. And allele frequency differences among the fine-mapped variants and KIV2 length explains the variation in median LPA across populations, in particular between Europeans and Africans. And the LPA type 2 diabetes association appears to reflect reverse causation. So reminding you of everything you've learned today, VNTR alleles can be estimated from whole-exome sequencing depth of coverage, and estimates can be refined by pooling measurements across shared haplotypes. Coding VNTRs at LPA, ACAN, TENT5, MUC1, and TCHH are strongly linked to diverse human phenotypes. And as Mirtash already advertised, my talk for next year will be about the phenotypic impact for non-coding VNTRs. And with that, I'll just like to thank my co-authors and the funding agencies, and thanks.